On the cloud. Okay, so we're going to start. We had a little technical problem. So we already discussed a bit about the objectives of this course, which are understanding basic concepts and get familiar with the data visualization methods that we have available. And then we're going to talk a bit about the major limitations of this type of studies, so, which is very important to understand. And that's what I expect from you during our weekly classes. I want you to bring me papers and tell me, I think this is a study that they did this, but they didn't do that, or they use this type of techniques that how we know has this and that limitations. Okay, so this is the objectives of this class today. And we're going to start with a little introduction about why everybody's talking about the microbiome and bacteria analysis. So that's what we're going to see right now. Like I said, there was a huge increase of the number of publications over the last years, and that's because of the release or the development of new DNA sequencing technologies, which allow now researchers to do that type of studies in any regular laboratories. All right, so just to put us in perspective, you will see, if we stop to think, our genome, it's much closer to the corn genome when you compare any two species of bacteria. And why do I like this slide? It's because it shows us that this world is really taken by bacteria. Okay, they have been here many billions of years before us. So we developed, we evolved in this world along with these bacteria that are literally everywhere. Okay, and including in and on us. So I'm pretty sure everybody already heard about the sentence that we are 10% humans and 90% bacteria, because we carry 10 times more bacteria than human cells, which make us, a super organism that is really all of the time interacting between the host and the microbiota. Okay, and these bacteria, they have a genetic capacity 100 times bigger than ours, which a huge metabolic potential, that it's able to take drugs that we ingest and just metabolize them before even we can do it, or they are producing a huge amount of neurotransmitters that will then be absorbed and will stimulate the nervous system or the enteric nervous system. And this has a direct communication channel with the brain. That's what we call the gut-brain axis. So this bacteria is very important for us to our homeostasis. And just to be, this is a bit sensationalist, but they have maybe, or they did recalculate this number of bacteria, and some people claim that it's not really 10% of human cells, but it's more like 50%. So we carry the double of bacteria than human cells, which is still, it's astonishing amount of bacteria. And the majority of these bacteria are in the intestine, and that's why we give so much attention to the intestinal microbiota. Okay, and as you know, the government has been given millions and millions of dollars to perform microbiota studies, in which we are using those new technologies to characterize the microbiota at the different parts of our body, and to understand the role of each of uh, those microbiotas and the consequences of changes in microbiota. So we now know that this is like very, very important for us. And the question is, what has changed over time, and why just now, we suddenly became very interested in the microbiota again. And as you know from school, the methods that we used to use were bacterial culture. But what scientists already knew at the time is that when you look under the microscope, they could see that only between 5 to 10% of bacteria could actually grow under those traditional culturing conditions. And therefore, the vast majority of bacteria, they became unknown until recently, when we developed those DNA sequencing technologies. Okay, so the bacteria might not grow on the culturing plate, but the DNA is still there. And therefore, if you go there and just sequence their DNA, you have a much broader picture of the composition of that microbiota. And over the last decades, those technologies have become much more available for us. They became smaller, faster, cheaper, easier to use, and to the point that it has really been considered a revolution in science. And there are many different companies that offer services for microbiota analysis, um, including this science, citizen science-based project that you pay just the price of sequencing around $200, and then they're gonna send you the analysis of your microbiota. You send a sample by mail, and and then they are able to use the data that they collect for the science. And they started now to publish many studies that are available in their website if you're curious to check. Uh, but the first technologies that were using DNA sequencing, they were, like I said, very laborious and very slow and expensive. To give you an idea, the first method developed by Frederick Sanger, which was 2 times Nobel Prize awarded for chemistry, the technology he developed was, he was sequencing, or he was finding one base pair at a time. To give you an idea, it took almost 20 years to finish the whole analysis of the human genome, and it costed around $3 billion to run that project. And right now we can do the same type of analysis by $2,000 or $3,000 in a matter of 2 or 3 days. Okay, so it has really developed a lot. And like I was discussing in the class we gave last week, these technologies are still evolving, and in a speed that is just incredible. And probably in the next 10 or so years, this is going to become a routine test that we can do, just like we run PCRs nowadays. We're probably going to be running microbiota analysis. Okay, so yes, I think it's very important to understand everything and to be working in this field, because very likely you're going to be reading studies if not publishing, I hope, studies about microbiota. And I wish and I hope all of you have read this publication, in which we try to explain in an easy language those basic concepts. So by now you all should be familiarized with those methods. And if you haven't read this, I recommend you to, go over and over, and if not, memorize this paper, then in a way that we can carry on our discussions in a dynamic and easier way. So everybody should know everything it's in there. And if you have any questions or doubts, go back to the paper and read it over and over, okay? All right, let's start a bit with some definitions. And there is a lot of mistakes in the literature. We use those terms very undistinguished way. So let's start in theory what microbiota refers to, which is the collection of all the microorganisms in an environment. So if you take all the bacteria, archaea, viruses, and small parasites like protozoas all together, and you are going to have the microbiota of that environment. So if you are talking about the rumen microbiota of the cow, that means all the fungi. We are missing fungi here. Yeah, it's small eukaryotes. Okay. So like we are talking about all the fungi, bacteria, viruses, everything that is living in that rumen, that comprises the microbiota. If we are talking about the, I don't know, my coffee microbiota, there is a lot of bacteria here. So everything that is in here is really the coffee microbiota, okay? But we normally study the bacteria because they are the vast majority. We think, or we want to believe, that they are the most important organisms in the environment, in that environment. So we use this term microbiota as a synonymous to bacterial microbiota. And once in a while, when I submit papers for publication, I'm gonna get a reviewer saying, look, you are not talking about microbiota because microbiota refers to all the microorganisms. You are really talking about bacterial microbiota. And then I say, okay, let's change everything, and let's say bacteria instead of microbiota, which would be more adequate. But in real life, we just use microbiota to refer to bacterial microbiota. Is that clear to everybody? Okay, I see. Yes. Good.
Yes.
Good. Now, I'm also sure that all of you have heard about genome. And the genome is the collection of genes of a organism, okay? So if I'm talking about the human genome, I'm talking about all the genes that are inside of my cells, okay? If I'm talking about the E. coli genome, I'm talking about all the genes that are inside, all the DNA that is inside of the E. coli, okay? So it's really clear, really easy, but what we have done in the last years is to combine those two terms, microbiota and genome, and then we come up with what we call the microbiome, okay? And this is already a source of confusion because a lot of people started using microbiome as a synonymous to microbiota. But in theory, it's wrong. The concept of microbiome It's the collection of genes that we have in the environment, the collection of the genes of the microbiota that are present in an environment, okay? All of the genes, all of the genes. So if we are doing taxonomic studies, meaning that we are investigating which bacteria is there, and therefore we are just sequencing a one single gene or a piece of the gene that's going to tell you the taxonomic classification, it's going to tell us the name of the bacteria only, then we should use the term microbiota. But if we are doing sequencing of all of the genes, not only taxonomic genes, but all of the genes, all of the metabolic profile, then we're gonna use the term microbiome, okay? Again, in real life, people use microbiome as a synonymous to microbiota. At the beginning, when I was reviewing papers, I was a bit picky with that, and I would say, you are not evaluating the microbiome, you are evaluating the microbiota. So please change the terminology. And, because I would say 95% of the studies, they are really just evaluating the microbiota and not the microbiome. But, I start to be a bit more flexible and relaxed because people said, yeah, everybody uses microbiome. And I think it's more appealing, and people, are just trying to sell better their studies. And I said, okay, I can live with that. Now I don't care too much about what they say, but I want all of you to be aware that when we evaluate taxonomic genes only, we should say microbiota or bacterial microbiota. And when we do what we call shotgun sequencing, then we are evaluating all of the types of genes, and therefore we use the term microbiome. Okay? Yes? Melanie? Good? Are you awake? I'm teasing you.
Yes, it's okay.
Good. All right. So, and now we also want to use or to define the term population, which refers to one specific type of bacteria or species of bacteria. And therefore, if we are talking about, for example, Salmonella, then it's a population within a community, okay? So if I'm talking about E. coli, it's a population of bacteria inside of a community. If I'm talking about Prevotella, it's another population. So I can have, in this example I gave you, 3 different populations, all of them inside a community. And therefore, when you have many populations together, we have what we call a bacterial community. Okay? This is just a detail, but when I'm correcting a paper, and I learned that the hard way, I learned that from probably some biologists correcting me during the reviewing process, and people saying, you are using the wrong terminology. You are not talking about a bacterial population. Here you are talking about a community. So it's important when you write a scientific paper to use the right terminology, so the reviewer will get not even a paper, but a proposal, for example. Sometimes I review a proposal and I say, it hurts a bit, to read when it's wrong, but it's not like really the results of your study is the most important, but it's always nice to speak a right language. It's right when you speak in English. I'm sure I'm making a lot of mistakes because I'm not a native speaker. So if a native speaker is listening to me, it hurts to all the accent and the mistakes. But the same when you're writing a scientific article, you should use the correct language and terminology, okay? Yes, don't use flora, because flora, it's good for people to understand because it's a common term used in the media, you know, the microflora, the gut flora, but in theory, flora refers to plants. So we are not talking about plants here, we are talking about bacteria. Is the zoom window in the front of my slide, or you can see the whole slide?
We can see the whole slide.
Okay, you can see the title well? Yeah, okay, good. So let's start talking about the concepts. What is richness? And here I can get help from a student that is willing to open the microphone and tell me the definition of richness. Don't fight, one at a time.
It's the number of species, number of different species.
Yes, good. So the richness is the most simple measure of or index, I can say, that we use to characterize a community, okay? It's really the number of different species or the number of different populations that we have in the environment. So if I ask Sarah, who is Brazilian, to go to the Amazon forest and count the number of different species of trees that are in Amazon, and she comes back and tells me, well, I count 1,000 different species of trees there. I could find 1,000 of different species. And then I'm gonna ask you, what's the richness of trees in Amazon? That's a question for you. 1,000. Good, good. So it's very simple, right? If I tell you that there are 735 different species of bacteria in the rumen of a cow, then the richness of that environment is whatever number I just said, I forgot.
735.
Yes. Okay, good. So it's very easy to understand. And now we're gonna do a little exercise here. And I'm gonna ask you, what's the richness of community A? How many different populations of bacteria do we have there?
Five.
Five, right? So I can call my 9-year-old kid here, or even the younger one, and I ask him and he's gonna say 5. So I guess everybody is good with that. And now it's very clear also that community B has the same number of bacteria. They also have 5 different populations, right? Yes. I guess everybody agrees with that. But you can say, well, if they both have the same richness, they are equal, they are identical. But now I want you to tell me, what's the richness of community A?
5.
5, okay. And what's the richness of community B?
5.
The same, right?
Yes.
But do you think they look like each other? Are they identical communities? Very clearly, they are not. So I'm gonna ask you, what's different between them?
It's the diversity.
Okay, the diversity is different, but before we get into the diversity concept, there is a more basic concept that is different between them.
Abundance.
Abundance of each population, which is?
Distribution. Evenness.
Yes, distribution or evenness. Okay, so here I introduce you the concept of evenness. Evennesses or evenness, yes. Whatever, however you pronounce that correctly, it is really the distribution of each population. You can see that here on the left, the green bacteria, the round green hairy bacteria, they are much more abundant. They are much more prevalent. The proportion of bacteria is much higher. Is that clear to everybody? You have, you can count, but let's say 80% of that community is comprised by this type of bacteria. And on the other community, on community B, what you can see is that they are much more evenly distributed, right? They are 20% each. So they have a relative abundance, or in this case, a absolute abundance of 20% and each, and therefore they are more well distributed, okay? Now we're gonna talk about diversity, which is not really a direct calculation, or it is, but instead of a basic concept, is just a mathematical formula, okay, that takes into account two different basic concepts, which is richness and evenness, okay? So whatever formula you use, and there are many different, I used to say that whenever a mathematician is bored and doesn't have anything better to do, he or she will go and develop a new diversity formula, because that, like I said, there are many, and what they have in common is that all of them take into account in the formula, they take into account the richness and the evenness, okay? And the final result is going to give us what we call diversity. Okay? So let's come back to our example here to explain you what the diversity is. Like somebody said, the diversity of those two communities are different. Let's say we are going to evaluate or to calculate the Simpson index, which is one of the most used index to calculate diversity. And to translate that, it is really just the probability. And I make a joke here to help students to memorize that. Let's say you have two bags full of poop, okay? And then you put your hand inside of the first bag and you pull out one bacteria from there. And then if I ask you, what's the probability that if you stick your hand inside of a bag full of poop, you take two different species or two different populations in a row, what's the probability that you're going to take a different bacteria every time? And this is what diversity means, or at least the Simpsons diversity means, okay? So if you put your hand inside of bag A, what's the probability that you're going to take the green hairy bacteria twice in a row? It's much higher than if you put your hand inside of bag 2 or B, right? Why? Because there are many more green bacteria in community A compared to community B. Is that clear to everybody? So in other words, if you put your hand inside of poop B, the probability you get a different bacteria is much higher than in community A, and therefore the diversity of community B is much higher than in community A. Is that clear to everybody? Yes, okay, good. I see you are awake. All right, now let's consider those two communities here. Here it's very clear that they have the same richness, agree? Because they all have 5 different species. They have the same, they have different evenness, and therefore they have different diversities. Now, let's see. I'm sorry about the noise. The kids are starting to speak up. I wanted to compare those two communities. What's the richness of community A and what's the richness of community B?
4 and 4.
Both of them 4.
4 and 4, and therefore they have the same richness. But what's different between them?
The population.
They have what? Yes, they have what?
Different population.
Different populations. Do they share any bacteria in common?
1. Yes.
Okay, so they share one type of bacteria, which is the green bacteria, but the rest of the bacteria is completely different, right? So what's different between them? Which concept is we use to classify them and to say there is a difference between those two communities?
Membership.
Yes, membership. Okay, so the membership of those two communities is different. Okay, meaning that the composition is different. They only share one type of bacteria, but in order to have the same membership or exactly the same membership, they should be identical. Okay? Is that clear? Now, keep looking at our community and tell me about the membership of those two communities, A and B. What do you think about their membership? Is it the same or different?
Same.
The same.
Same membership.
Same.
Now keep looking at them. This okay, so this should be the same slide. They have the same membership. What about now? What's the membership between them?
Yes, the same. The membership.
It's the same.
Do you agree, Sarah?
Yes, it's the same.
The other Sarah Argehani?
Yes, it's the same.
The same membership, right? But they look completely different, don't they?
Yes.
Yes. So what's different between them?
Diversity.
The diversity, even the diversity could be the same if they have the same richness and the same evenness. Let's say there is exactly the same number of red spiky bacteria on the red than the green hairy on the left, then they would have the same diversity. Okay? So they could have the same diversity, but they still look completely different. What is different between those two communities? If they have the same membership, they have the same diversity, the same richness, but they are still different. Which concept we can use to distinguish them?
Structure.
Structure. Okay, very good. So they have a different structure. And therefore, if the membership takes into account composition, the structure takes into account the composition and the distribution of each population. So it takes into account membership and the evenness of each bacteria. Is this clear to everybody? And this is really when normally I see all the students saying, yes, it's very clear, and it is because it's easy. It's easy to understand, but if you don't practice in your head those concepts, they're gonna fade away and you're gonna forget it, okay?
Yes.
Maybe tomorrow you will remember that, but next week you're gonna think, wait a second, I remember richness, I remember evenness, diversity, oh yes, it's the formula. And then when you start to add membership and structure, then you forgot. What's membership, what's structure again? So if that happens, you go back to your notes. I can share my slides with you. You go back to maybe the recording, or you go back to the basic concepts paper, and you have to refresh that into your mind constantly in order to not forget, okay? All right, so I'm gonna give you a last example about two different communities that they really look like each other, and they have the same structure. Why? Because the vast majority of bacteria, they are the same. They share the same type of bacteria, which is the green round bug. But what's different between them?
Membership.
The membership, it's completely different, right? They only share one type of bacteria, and the rest of the bacteria, it's completely different. So they have the same structure, but they have a different membership. And you're gonna see many situations in which you do the analysis and they have the same structure, but the membership is different. And what's really changing is what? Rebecca, what's changing between them?
The rare bacteria.
The rare bacteria. And I'm asking Rebecca because she saw something like that in her results. And when you have a change in the low abundance bacteria, then it's when you have the same structure, but a completely different membership. Okay? Good. So I hope this is clear. Just to make things a bit more complicated, you can also assign taxonomy into your analysis. You can give it a weight to the genetic similarity between the bacteria present in a community. And the example I normally use is the similarity between Escherichia and Shigella. So let's say you have a community that is comprised mostly by Escherichia and Shigella, and therefore they are, let's say they are cousins. And when you compare that genetic similarity with other species, you know, so they are bacteria, and let's say if the others are firmicutes or Bacteroidetes, you can assign a weight to that genetic similarity, and therefore you can have a different type of analysis that we call taxonomy. Okay. All right, let's make things more complicated. It can always get more complicated. Now we're gonna see people talking about alpha diversity and beta diversity and what they refer to. Okay, so the alpha diversity is the characteristics of each community. When you get, let's say, my microbiota, and I'm going to analyze that. I'm going to count how many different bacteria are there, therefore the richness. I'm going to see how well distributed they are, and therefore evenness. And then I'm going to get those two numbers and combine them into a diversity index, and therefore calculate diversity. And I'm only talking about my microbiota. So I'm characterizing the community of one single sample. So I'm talking about alpha diversity measurements, alpha diversity indices. Is that clear to everybody? Okay, good. And now, if I do the same with the microbiota of Ali, I have all the measurements of his microbiota, and I want to compare with my microbiota. Okay? Then what I'm doing is a beta diversity analysis. When I'm comparing communities, I have a beta diversity analysis. So if I'm comparing membership between them, if I'm comparing structure between two different communities, I'm doing a beta diversity analysis. Okay? What is important here is not confound, not to mess up diversity indices, which are mathematical formulas that take into account richness and evenness, and that are alpha diversity measurements. So people normally, and that happens so often. Just last week, we got a paper for our special edition in Equine Microbiota. And they had a graphic, a figure with figure 1A saying alpha diversity, figure 1B saying richness, and figure the other figure was evenness. And I say, this is wrong. All of them are alpha diversity. And then I came back to the material methods, and I saw what they did was Simpson, Shaw, which is a measure of richness, and Shannon, which is a measure of evenness. And I say, you cannot say alpha diversity, richness, and evenness. You have to say diversity, richness, and evenness. Okay, because all of them are alpha diversity indices or measurements. So this is wrong. And people really mess up alpha diversity, beta diversity, and diversity indices because it's the same word, right? It's diversity, but one is alpha diversity, another is beta diversity. And you have the diversity indices, which are Simpson, Shannon, and many others. Okay? So maybe I'm gonna pause the recording here to get questions. All right, let's see how far we can go today, and if we, like I said, I have a meeting at 10. If we have to stop, we're gonna finish next week, okay? So how can we visualize the analysis of all those concepts that we just discussed?
We are not seeing your slides again.
No?
No, sorry, no, yes, methods for, yeah.
Yeah, methods for data visualization. Tell me if it changed now.
Yeah.
Yes?
Yeah.
Okay. So when I'm teaching that in person, normally what I do is to give these results and tell students. It's a little practical exercise. So I tell them, I just finished the analysis of two samples, sample A and sample B. And here are the results. Okay, sample A, 80% of the reads, they were classified as firmicutes, which is a phylum of bacteria. And in sample B, only 60% were firmicutes, but sample B had much more Bacteroidetes and less Bacteroidetes in sample A. And here is the final result. Let's say I had 100 reads, 100 DNA reads in each sample, meaning that 80 of those reads were classified as firmicutes in sample A. 10 of those reads were Bacteroidetes, 5 were Proteobacteria, and 5 were Verrucomicrobium. Okay? Then I tell students, go there in Excel and bring me back a plot that represents those two different communities. And some people will come back with those pizza discs, like 80%, 10%, just like regular disc plots, but the majority will make column plots or something like that, which is really clear. It's a very good way to visualize data. All right, so this is what normally we see. The column plots comparing relative abundance is really the most used in microbiota papers because it's very nice. You can compare side by side two different samples, and it's very easy to visualize the differences. Okay, so in this example we use, we have 80% of firmicutes in sample A, but sample B has many more Bacteroidetes. Good? All right. So here are some real-life examples. This is the comparison of the microbiota of 6 healthy horses. So these first 6 are healthy and the rest are horses with colitis. And what we can see, or diarrhea, what we can see here is that healthy horses, they have more, in general, more firmicutes, and horses with diarrhea, they have more Bacteroidetes. Okay, and the other good thing about this type of graphic representation is that you can see individual variations. For example, you can calculate the average of those relative abundances and make, let's say, 2 different plots. You can say that the average of group 1 was 80% firmicutes, and average of group B was 40% Bacteroidetes. But in here, when you are expressing averages, you cannot see individual variation, right? So every time I see, I review a paper showing averages of relative abundances, my first comment is, I cannot see data variation. You have to it would be nice to see, even as a supplementary file, if you can do something like that. Okay, make it a plot with every single individual in that plot that we can see. For example, there was one individual here that had a lot of fusobacteria, right? Almost 40% of its bacteria were fusobacteria. Why is that? Other animals had just a little bit, right? Some had none. And why is this animal having a lot of fusobacteria? If you merge everything into an average, you're gonna see that 2% of horses with diarrhea had fusobacteria, but that's not true. This one had 0, okay? Why you are saying that they have 2 or 4% if they had 0? And this one, it didn't have 2%, it had 40%. So when you put one side by side, you can clearly see individual variation. For example, those 2 animals here, they were healthy, but they had a very high abundance of Proteobacteria. And why is that? Interestingly, those two horses, they were housed in the same facility, which was a teaching hospital in Guelph. They were teaching horses. And they were housed together, and it's very interesting because they have the same proportion of bacteria, okay? But they were different from the 4 horses that were housed in farms, okay? So that's one way to analyze or to visualize relative abundance. Here's another example. Here is the relative abundance of the stomach, of the bacteria in the stomach of horses. And you can see very easily here that there are 2 different types of samples, right? One with high amounts of firmicutes and another one with high amounts of proteobacteria. And it's very interesting because in this study was one of the first, and what they were doing was to just, descriptive study. But these 7 or 6 first horses here, 7, they collect the samples by endoscope in horses that have, like donation horses that have been in the hospital for a few days, and they kept them off feed, no food, for 1 or 2 days, and then they went there with the endoscope and collect samples. But those others here, what happened was they were also donations, but they went straight to euthanasia, and they collect the sample directly from the stomach. So you can see that 2 different methods can yield completely different results. And here in this type of analysis, in the column, you can visualize that it's so clear that, and you can compare the different proportions of the bacteria or relative abundances of bacteria. Good. All right, so another example, what you can do, instead of showing each column, you can show the area. Okay, in this example here, we have the microbiota from children from, they were comparing microbiota from children in African tribes that live, like we used to live during the agricultural revolution. So their diet is basically carbohydrates. Eventually they eat a bit of meat, protein, but then they compare it with the Italian children that had a very high fat diet. And they could see, this is the relative abundance of Italian children, that they have much more firmicutes. And here the African children, they had many more or much more bacteroidetes. What was interesting, that there was a group here of children that was comprised from Italian and African children, and they have a very similar microbiota despite being in different places. And they were very high in actinobacteria. And why do you think they were sharing the same microbiota if they were so distinct? Does anybody have a hypothesis for that? They were very young. They were babies. They were on breastfeeding. Okay, so if they were nursing from their mothers, and therefore it doesn't matter where they came from, but the diet was the same, and therefore they had the same type of bacteria. Okay, so this plot can tell us a lot just by doing relative abundances, and it could be columns, different columns. But for example, this plot is from Rebecca's data, and it's comparing the microbiota of horses before and after fecal microbiota transplantation. Like just eyeballing, you can see that there is a group here that has more fibrobacter. Though this donor here had more, what's the green one? subdivision 5, verrucum microbia. So it's very easy to look and identify and compare samples, okay? And what is this plot? This is my garden. After a day of snow, I opened the window and I said, oh, I see a relative abundance area plot. And my wife said, you are working too much. You slow down. But now, one limitation also, like I said, when you report relative abundance plots is that you don't have, if you are reporting the average, you lose the individual variation. But if you are reporting individual variation, you lose the capacity to show a lot of data. Okay, let's go back. For example, here we are talking about the 10 most abundant bacteria, but let's say I'm talking about those here that are not as abundant, or even the ones that have lower abundance. When I compare them, the problem is that I cannot visualize them here. Okay, so one way to bypass this problem is to compare them individually. Okay, I'm sorry, just give me 1 second. No, you can close the door, please. All right, so okay, so you can compare each type of each population, okay, in the different groups. For example, here into her preparation of fecal microbiota transplantation, we were comparing 2 different methods of preparing. And you can see here that one type of preparation had many or much more Escherichia than the other type, okay? So you can just compare each individual, the relative abundance of each bacteria in each individual, and you can compare groups. You can just break up that busy column type of representation and do that into this type of, I forgot the name of this analysis, dot or blocks plot, right? Okay, the other way that everybody is using now, it's everybody likes it's what they call violin plots. You can show here the different populations. You can compare them between groups. You can express the average and also data variation. Okay, you can express how variable was this data. So it's easy to see that the mean here or the average was very different, but you can also see that many of those samples, and here it's the relative abundance of, I'm sorry, it's in Portuguese, the title, the relative abundance of Staphylococcus in milk samples from that study I mentioned, milk samples from the tank of farms with low somatic cell count and high somatic cell count. So there was a statistical difference, but you can see here that many of those samples had the same relative abundance as the low somatic cell counts. So it's just another way to represent the relative abundance, okay? Now, I'm glad we could make up to here because now we're gonna start talking about principal coordinate analysis, which is a measurement of or a representation of beta diversity, meaning that we are comparing 2 different communities, okay? So in order to start, we need to do what we call a contingency table, which is a very simple table in which we're gonna put or insert the number of reads or the relative abundance of each bacteria for each of the samples, okay? Here, I was using this example last week, so I kept the names. I have my microbiota here, and here are the different types of bacteria I'm analyzing, okay? So let's say I had 100 of my reads. They were classified as the green hairy bug. 100 were the red spiky, and so forth. And then I had Dr. Toribio, Ramiro Toribio microbiota analyzed, and we could see that he had 200 reads classified as this one, 200 as this one, and this is another professor, Diego Gomez, that were present. So it's basically a relative abundance analysis, but without the plot, right? Is this clear to everybody?
Mhm.
So what we do now, we get this table, okay? And we do some fancy mathematical formulas to transform that, which I'm not gonna get into detail. But just looking at here, it's very easy. You can see that, for example, we call, let's say the green hairy bacteria, we call it OTU1, and OTU stands for Operational Taxonomic Unit. Don't worry about that. It's just one type of species, okay? So we have species 1, species 2, species 3, and species 4. Just looking at this plot here, or at this table, I'm gonna ask you, which microbiota are more similar? Just looking here, I had 4 bacteria that were classified as species 1, and Diego had 3 bacteria that were classified as species 1, but Ramiro had 9. Okay, I'm asking you now, which bacteria is more similar to mine, Ramiro or Diego's?
Diego.
Diego's, right? It's easy to see that. So if I have to represent that in a plot, in a principal coordinate analysis plot, which bacteria will be together? Which microbiota will be together? Like you said, mine and Diego's will be together, and Ramiro's will be very different, right? So this is what the principal coordinate analysis does. It brings all this data into one dot, okay? It makes it look like one dot. And then what you can see is that 2 dots that have similar microbiota, meaning they are more similar, they're gonna be together. But the dot that has a completely different microbiota, it's gonna be very far apart. And this needs to be clear to everybody. You're gonna see this type of analysis in every single paper. PCOA. Go ahead, Melanie. Yes.
I have a question, Marcio.
Yes.
What means those numbers on each of each axis?
Oh, yeah, that's a good question. Everybody asks that question. We do, so when you do the matrix analysis of those tables, you end up with something that we say it's multidimensional or highly dimensional number, which cannot be represented in a graphic that the human brain can visualize, can understand. We can only see up to 3 dimensions, right? 3D. Mm-hmm. When you go to the movie theater, that is what they call 4D or 4 dimension, the fourth dimension is not really something that you can see, but you can feel. So the seat will shake, you know, and it will twist you. So this is the fourth dimension, which is not really a dimension, it's just a marketing, because the maximum you can see is the move theater screen, which has 2 dimensions, and the deep sensation or deepness. I don't know how to say that in English, but you can see things, you know, in a more depth, different depth. So you can see things coming towards you. So this is the third dimension. We cannot see a fourth dimension. Okay. So we do what we call vector analysis. There is a German name, I can't remember the name, to reduce this highly dimensional number into 2 or 3 dimensions. And this is where those values come from, which are basically distances that there is no unit. There is no centimeters, it's not percentage. It's just a number that the analysis will yield. And if you include another samples here that will be even more distant, these numbers will shrink so that everything can fit there. And then you're gonna have different numbers, but it doesn't matter. Then the numbers here never matter. What matter is that if you see here 0.2, there is probably 0.1 distance between those 2 samples, and if you calculate here, it's going to be probably 0.6, the distance between them. So you can just compare distances, okay? But there is not a unit that you can attribute to that. Okay.
Okay. And does it tell us something when we are in minus, or we only see the distance? Okay.
Just a distance.
Okay.
It gives you the 0 to give you a reference that you can. Normally, you're gonna see other examples that you have the axis represented here, but the 0 doesn't mean it's negative, doesn't mean it's good, it's bad, it doesn't mean anything. It just means that from here to here you have 0.4, and from here to here you have 0.3. So if you want to know the distance between those 2, it's going to be 0.7 whatever unit. It's just bigger than this distance here, which is 0.1. Okay. What else? Yeah, and this, because this is I think it's Euclidean type of plot in which you can have, it's like a GPS. Okay, where are you? Oh, I am in, what's the coordinates of Saint-Hyacinthe? You have the latitude and the longitude. So you have 2 different coordinates that can tell where you are exactly. And then you want to compare where Rejean is. So Rejean, let's say he doesn't live in Saint-Hyacinthe, he lives in Saint-Aubert, then he's going to have other references compared to the 0 here. So you can compare both of them. And now Rebecca, she's even further. She's in Montreal. So the longitude here is 0, but the latitude is 0.4, minus 0.4. See, so you can just determine where the dots are in relation to the 0, to the ground mark here. Okay?
Okay, thank you.
Good questions. Any other question? Because this is important. Now I'm gonna give you other examples. So here is the representation of those 6 healthy horses that I compared the microbiota with the 10 horses with diarrhea. And as you can see here, you can find a cluster comprised by the microbiota of healthy horses, okay? And just if I had to guess, I would say that those two horses with high amount of, I think, actinobacteria that were different from the others, they should be together, okay? So they should be side by side. And that single horse that had a lot of fusobacteria had a very different microbiota. So I would guess that it would be this sample over here. Can everybody see the arrow of my pointer?
Yeah.
So see, this is the graphic representation, but now I'm gonna see for one of my senior students. Can you tell me the difference between relative abundance when I'm comparing relative abundance and when I'm comparing a principal coordinate analysis? The most or the biggest difference, what the principal coordinate analysis is showing us or is taking into consideration that the relative abundance is not.
The distribution, the structure.
Yeah, the
The
structure.
structure.
I cannot see or I cannot tell if this principal coordinate analysis is comparing membership or structure. What do you see in a relative abundance plot? Let's say at the genus level, what I can see at Rebecca's data, how many different bacteria there?
In the PCOA,
In the relative abundance plot.
okay.
The
How many?
abundance.
The richness and the membership.
No.
Richness and
How many different bacteria I was presenting at Rebecca's plot? Let's go back there.
A lot.
A lot. All right, here. How many bacteria you can see here in the legend?
14?
15.
13.
Yeah, between 10 to 15. I can put 20 here.
13.
Okay. Do you think that in the fecal microbiota of those horses, I had only 13 bacteria, different bacteria? Was the richness 13? What's the richness of a fecal sample of a horse? What would you expect to be? Around more than 500, right? So anything between 700 and 1,000 different species of bacteria should be present there. How can you represent 1,000 species of bacteria in a relative abundance plot? It's just impossible. You cannot read the names, you cannot see. If here is already difficult to see the relative abundance, many of those bacteria, they will have a relative abundance, or I would say that 90% of that bacteria will have a relative abundance lower than 1%. So it's just impossible to take everybody into account here. So what we are looking here is just the most abundant bacteria. Is that clear? Now, Rebecca, if you look at this table, we have only 5 species. Here, only 4 species. But really, in reality, what this analysis does is to take every single bacteria that you found in your analysis. It's a huge table. It takes a lot of mathematical effort to calculate that so that we can have a computer. I just make that simple for you to understand. But how many of those bacteria, like I said, this analysis is taking into consideration?
All of them.
All of them. Okay. So the difference between looking at a relative abundance plot and looking at this is that here we take all of the bacteria into account. Is that clear? Therefore, in the relative abundance, we can compare the similarity between the most abundant, but here we can, in fact, compare the similarity between, the true similarity between those communities. Okay?
Mhm.
Yes.
Here is another example. We were evaluating the microbiota of broiler chickens, that each group was receiving a different type of antibiotic. Okay, so here you have the controls that receive no antibiotics. You can see a clustering of the blue dots here, and when you compare, for example, with animals receiving avilamycin, the antibiotic was really shaping the sickle bacteria in a way that was statistically different from the controls. And the same happened to bacitracin, for example, enramycin and virginamycin, but not with this other antibiotic that you can see that it's just overlapping the controls, and therefore there was no difference in no statistical difference between controls and this less antibiotic. Okay, so you can see, you can find the clusters. I can break that into different plots to compare each antibiotic. For example, in this is a 3D analysis that we were comparing the microbiota of the different intestinal tract of horses. So in red, you have the dots from the stomach bacteria. In pinkish, you have the duodenum. Yellow was the ileum. And here, as you move towards the distal gut, you can see that they start to cluster in here. So therefore, the bacteria of the distal gut from the large colon, the small colon, rectum, and feces, they were very similar between them. Okay, so you can clearly see a big difference. You can find clusters, and just to finalize the question from Melanie, we have 5 minutes before we finish. What I wanted to say here that is very important for you to understand is that when you do a principal coordinate analysis, you're gonna have two axes here, but it really gives you all the different axes, and we're just gonna represent the 2 or the 3 first. Okay, so here we are representing the 2 first axes or components or coordinates, and here we are representing the 3 first coordinates. Okay, and when you do the analysis, and we're gonna get into more details when we do the workshop this summer, you're gonna see that it gives you a number. Okay, so when you compare, when you do the first analysis, it's gonna tell you, let's say 50%, meaning that if you put all the samples in a straight line, this is gonna explain 50% of data variation. So you're gonna have samples from stomach here, you're gonna have samples from the duodenum here in the line, and you're gonna have samples from the feces and distal gut very far in that line. But if you add another axis, now you have 2 axes, then you're gonna see something like that, right? You're gonna see one on the top left, one on the right bottom, and then you're gonna have the number of the second coordinate. So the first one was 50, the second one was 10. So that means if you put all the data together in a bidimensional plot, this plot is going to explain you 60% of data variation. Okay? But there will be situations in which this data variation, the explanation is very low. Let's say the first coordinate explains you only 20%. The second coordinate explains you only 10%. So 20 plus 10 altogether, this is gonna be 30%. Okay, and then you're gonna say, oh my God, I gave fecal transplant for those horses, but I cannot see a difference. Why is that? So I want you to think that I have two round things here, okay? That makes it smile. So I have two dots here. And if you are looking at my screen, you are only seeing a two-dimensional image, all right? Let's exaggerate. Let's say it's like that, okay? So this variation of this data is explaining only 30% of the variation, but they are very close to each other. Don't you agree? Let's put it like this. They are very close to each other. But if you look, if you add a third dimension, you're gonna see that one is further and the other one is behind, and therefore they're not as close as they look like, but they are actually, if you add a third dimension, you're gonna see that they are very far from each other. Is that clear?
Yeah.
So sometimes you're gonna do a analysis like that, or like the horses, and you're gonna see that the two dimensions already explain 80% of data variation. It's fine. You don't have to add another one. And like Patrick Sloss was said in his workshop the other day, the human eye can only see two dimensions. If you can play with if you can play with the file in a way that you can twist it and turn it, there are softwares you can do that, then it's fine to add a third dimension or something like that, then you can still visualize, you can imagine the data variation. It's fine to use a three-dimensional plot, but it's just the three first coordinates, if you put them all together, the third one will explain more of the data variation because you are adding another dimension to your analysis. Is that clear to everybody?
Yes.
All right, so we have to stop here. I want you, to go. I can send you the slides, and you review that, and you can bring me questions for the next class.